Hello, and welcome to this e-seminar which will summarise the content of FSA Report FS 900246, a review of Allergen Analytical Testing Methodologies, which was undertaken and produced by Fera Science Ltd. This e-seminar has been produced as part of the Joint Knowledge Transfer Framework for Food Standards and Food Safety Analysis, and which is funded by the Department for Environment, Food and Rural Affairs, the Government Chemist Programme for 2023 to 2026, the Department for Science, Innovation and Technology, formerly the Department for Business, Energy and Industrial Strategy, the Food Standards Agency and Food Standards Scotland. UK food labelling regulation require that the presence of any of the 14 priority allergens be stated if known to be present in a product. As food allergy is an important food safety issue, it is essential that there are validated methods in place to both detect and quantify the presence of allergens in food products. The current primary methods for screening for allergens in food products is ELISA, but with PCR and various mass spectrometry methods being introduced as additional confirmatory approaches where the result is unclear. However, the suitability of these methods varies considerably depending on both the allergen and matrix being tested. It should be noted that other countries may have different lists of allergens of concern. 13 of the 14 UK prescribed allergens may be detected by identifying the presence of a distinct protein, which is the primary focus of this presentation. However, as sulfur dioxide is not a protein, chemical methods are required to detect its presence. Enzyme-linked immunosorbent assays, or ELISA, is currently the primary method for detecting the presence of allergenic proteins in foods. The technique utilizes antibodies that have been produced to preferential bind to a specific protein, the allergen in this case, and are usually tagged with an enzyme which can be employed with a colorimetric reaction to provide a visual result. A variety of commercial ELISA kits are available which can be used to identify and quantify the presence of the majority of the 14 core allergens, either qualitatively or semi-quantitatively. In terms of ELISA testing, there are a number of differences approaches based on the type of antibody, but also reporter system employed. ELISA testing uses either monoclonal or polyclonal antibodies to bind the target protein. The use of polyclonal antibodies may be beneficial when testing processed foods because they can bind to multiple locations on the target protein and therefore their use increases the likelihood of detecting the desired proteins. However, because polyclonal antibodies do not target a specific protein, there is an increased risk of obtaining false positive results with kits that utilize them. Conversely, monoclonal antibodies target a specific protein and therefore may be used to distinguish between similar proteins and can help reduce the risk of cross-reactivity. Aside from the type of antibody employed, ELISA approaches differ in the reporter system they employ. In broad terms, these can be based on either a sandwich or competitive assay approach. Sandwich ELISA involves using two antibodies to capture the target protein, whereas competitive ELISA utilizes a secondary protein to competitively bind to the antibody and enable the quantification of the target protein. For example, when detecting gluten after treatment to break it down, any remaining gluten fragments may be too small to be detected quantitatively by a sandwich ELISA, and thus competitive ELISA should be used. However, competitive ELISA approaches typically utilize less rigorous extraction, which in turn may result in incomplete extraction, especially in processed matrices. Whilst ELISA is currently the primary method for allergen screening, it may be the subject of significant drawbacks. These can include extensive variation between test kits in terms of both the information provided with the kit and the results that are obtained. In this respect, certain kits fail to provide the relevant information for determining the suitability of the kit for the intended analysis. This can frequently mean that target protein is not specified, which may be problematic because allergens can be detected by multiple proteins form, some of which are only detectable in unprocessed food matrices. For example, ovalbumin and ovomucoid are allergenic egg proteins. However, whilst ovomucoid is unaffected by thermal processing, ovalbumin becomes undetectable. Therefore, prior knowledge of the specific protein targeted aids in suitable kit choice and avoids the risk of generating spurious results. As well as this, many kits do not indicate the matrices employed during for validation and whether these were raw, processed, or incurred, meaning individual labs have to expend resources ascertaining which kits are suitable for the intended analysis. Additionally, kits do not always state whether the LOD and LOQ were calculated using a buffer or an incurred food sample, even though the difference can alter the accuracy of the result obtained. Similarly, some kits give information about cross-reactivity when testing specific matrices, and others don't. This issue is exacerbated by kits constantly undergoing development, frequently under the same name, yet details about any changes are not always available. Assay cross-reactivity is a considerable limitation in allergen testing that can lead to inaccurate results. The risk of cross-reactivity with ELISA assays is a significant risk due to the similarities between the structure of target protein molecules in different species. Examples of foods which are recognized to cross-react include peanuts with chickpeas, almond with plum, peach with nectarine, pistachio with cashew, and hazelnut. Of considerable concern is the fact that mustard can cross-react with rapeseed, which itself is utilized in multiple food products, significantly obscuring the result. Additionally, as alternative protein sources for meat are developed, there is the risk they may cross-react with other proteins. For example, tropomyosin proteins in insects cross-react with tropomyosin in crustaceans and various mollusks. In addition to cross-reactivity causing false positive results, false negative results are also a problem with ELISA-based allergen testing. This scenario is frequently reported as the result of food processing. Typically, when an allergenic protein is subjected to heat, pH, and pressure changes, their structures may be altered so that they no longer bind to the antibodies being used to detect them. The result of this is that they frequently go undetected, with potentially significant consequences. The precision of ELISA allergen testing has been evaluated in the FSA report FS 900246 using proficiency testing PT data from over a 5-year period. The data from these proficiency testing schemes use data comparison to assess laboratories' capabilities to detect allergens in homogeneous spiked food samples. Following analysis of this pooled data set, it was determined that both the repeatability and reproducibility limits of the ELISA kit that were used had been reached, and that there was a general lack of agreement and repeatability between the different kits, including those from the same manufacturer. These findings also highlight that an improvement in performance has not been reported in over 13 years. Some specific examples of ELISA performance: the concentration of lupin detected and cross-reactives observed with other legumes vary between kits. Overall, fish can be difficult to detect accurately with ELISA, largely due to cross-reactivity and thermal processing. Mollusks seemingly increase in detectability by ELISA testing after processing. Conversely, milk presented only 2 to 10% recovery after processing. Gluten is well detected following thermal processing, although the matrix and source of gluten can affect the results significantly. For example, one study showed 100% recoveries with sausage, boiled beef, tomato sauce, and orange juice, and only 30% recovery for jam. Gliadin and glutenin are two allergenic target proteins in gluten. However, comparison is difficult as their units are hard to convert between. Casein, serum albumin, and whey are all milk allergens. However, whey has been seen to be significantly less thermally stable than the other two. Additionally, the production of some milk products involves fractionation, in which only some portions, e.g. casein, are included in the product. Therefore, it is essential that kits targeting the correct proteins are used. Differentiation between tropomyosin found in mollusks and crustaceans can be difficult. Therefore, specific monoclonal antibodies targeting the N-terminal region of the tropomyosin is necessary. A method using flow cytometry with competitive ELISA for the simultaneous detection of multiple allergens has been developed, with both peanut and crustaceans successfully detected at the reference dose. PCR is an alternative technique that may be used to confirm the detection of an allergen in some circumstances. PCR stands for polymerase chain reaction, which is an approach that permits the exponential amplification of low amounts of target DNA material to much higher and detectable levels in a short period using a thermal cyclic process. PCR may be employed quantitatively or qualitatively, and is primarily used in conjunction with ELISA testing as a confirmatory test. PCR may be used with most of the priority allergens, but it is not appropriate for all of them. For example, egg does not contain sufficient DNA to permit its detection without having to resort to advanced and complex PCR methods. In addition, it's not possible to differentiate between DNA from chicken egg and that from chicken meat. Similarly, the DNA from bovine tissue and milk cannot be distinguished, making confident detection of bovine milk with PCR difficult. Of particular importance is that PCR does not directly detect the allergenic protein, but rather implies its presence through detecting DNA from the organism of its origin. Therefore, care must be taken when making conclusions based on PCR results. For example, in some processed products containing sesame, the DNA is separated from the allergenic proteins. PCR is a highly versatile approach which is primarily used as confirmatory tests with allergen testing. As a confirmatory test, it is frequently used in cases involving fish or hazelnuts, and where PCR generally functions better following thermal processing than ELISA. With celery, PCR is currently the primary method for detection and has been reported to work even in incurred food samples. Additional examples of using PCR as a confirmatory test include being able to distinguish the allergenic almond proteins from many similar proteins, and where ELISA is recognized to cross-react. However, PCR can actually be less sensitive than ELISA, for example, in identifying the presence of mustard or hazelnut, but also in detecting the presence of crustaceans. Mass spectrometry is an emerging technique in the detection of allergenic proteins. Fundamentally, it measures the mass-to-charge ratios of ionized molecules to enable their identification. There are several different types of mass allergen testing, which is undertaken using liquid chromatography mass spectrometry, LC-MS, in which liquid chromatography is initially employed to separate out the peptides prior to injection into the mass spectrometer. MS-based methods that have been used include nano-LC ion trap, MS-MS, matrix-assisted laser desorption, ionization, time of flight, MS-MALDI-TOF, and single reaction monitoring, SRM-LC-MS. A benefit of using MS is the increased specificity of the peptide sequences identified, and therefore a reduced chance of cross-reactivity. However, MS methods require more development to achieve an increased sensitivity than is generally currently available, but also to permit the detection of allergens at the limit of detection and quantification possible with ELISA. While MS-based allergen testing has been in development during the last 2 decades, with methods now for use with eggs, peanuts, fish, and soybean, additional research is still required, for example, with optimizing an approach for the detection of mollusk-derived allergens. Mass spectrometry methods have been developed for most of the allergens, including multi-allergen tests. For example, mass spectrometry has been used to detect mustard in foods at an LOD of 0.25 parts per million, lupin at an LOD below the vital reference dose, and methods for soy, egg, and tree nuts have been developed with LODs similar to ELISA. However, current MS methods still require additional refinement to ensure they are sensitive enough to detect allergens at the recognized action levels. In addition, full validation of the methods across a range of different matrices remains to be addressed. Aside from the development of mass spectrometric methods, availability of an open-source protein database, along with an increased knowledge of food-related proteins, would be beneficial when developing new MS methods, particularly as the selection of target peptide or target peptides may affect assay sensitivity. For example, the peanut allergenic protein, ARA-H2, is a good choice of target for processed samples due to its thermal stability compared with other allergenic peanut proteins. The allergen sulfur dioxide occurs naturally in wines and is a preservative used in many foods and drinks. In terms of UK legislation, if it is present above 10 mg per kg, it must be declared on the label. Sulfites can readily be converted to sulfur dioxide and are therefore measured and expressed as sulfur dioxide. A frequently used detection method is that of aeration oxidation, AO, which involves freeing the sulfur dioxide from the sample matrix. However, applying this method for on-site testing by smaller manufacturers is frequently impracticable, as the procedure is specialized. Other, more efficient methods are also available. For example, iodic titration. However, this may give falsely high results due to interference from other components in the wine, such as ascorbic and erythorbic acid, or the colour compounds in red wine. Alternatively, spectrophotometric methods, enzymatic approaches, and an approach based on ion exclusion chromatography have all been employed. However, the Monier-Williams method is currently the most widely used for quantitative analysis of sulfites in both food and drinks, although there is a comprehensive list of ingredients which may cause false positive results. Regardless of the analytical approach adopted, key to any analysis is the fact that sulfur dioxide can be easily lost from a sample, and extra care must be taken when carrying out any of these methods, and special care given to quality control procedures. There are several areas within UK allergen testing that require additional development. Currently, other than for sulfur dioxide and gluten, there are no prescribed action levels for allergens in the UK. More auditing throughout the supply chain to reduce instances of undeclared allergens. This includes overseas suppliers of raw materials, as well as smaller sized businesses in the UK that tend to test less regularly. The development of multi-allergen testing would be beneficial in reducing manufacturers' time and expenses. Infrared spectroscopy methods can now successfully detect multiple allergens, but are not yet sensitive enough. Further method validation and education for food suppliers on how to choose appropriate test methods for their products. For example, some labs offer PCR testing for egg, despite it being completely ineffective if used on food containing egg white, as the yolk contains the only copy of DNA. Similarly, the low pH of tomato-based products can make DNA undetectable and thus PCR ineffective. Therefore, LC-MS methods can be preferable for confirmatory testing, but this is significantly more expensive. Additional method validation would also help analysis of difficult matrices. For example, in a trial, 12 labs attempted to detect sesame in mayonnaise, and only 2 were successful. more research into how processing affects the allergenicity of proteins has been suggested by Thrall. Finally, there is a need for greater transparency about the scale of allergen issues in the UK. Having a central database in which manufacturers publicise day-to-day challenges would achieve this. A significant hurdle in the reporting of finding from allergen testing is the interconversion of data types. In order to determine how well a test method and kit are performing, as well as deducing meaning from the results, some degree of data comparison is required. However, variation between buffers, calibration standards, antibodies, and target proteins used, even when testing for the same allergen, makes comparison of data between ELISA kits difficult. For example, data is often reported in terms of total milligram to kilogram allergenic protein, which can be difficult to convert to, and although various kits provide a conversion factor, many do not. Furthermore, MS data often relates to peptide mass, whereas PCR typically reports in terms of numbers of copies of genomic sequence present, and thus the conversion to total food allergen, whilst important for comparison of data, is extremely challenging. A survey on the allergen testing procedures of 7 UK food suppliers has been undertaken by Fera Science Ltd. The results showed that of the 7 companies surveyed, all of them test the finished products, 4 test raw materials, dependent on there being a recognised risk of allergens being present, and 4 test the production lines to validate their cleaning processes. In addition, one company also tests the rinse water prior to use for the potential presence of allergens. All 7 companies only test for the allergens that pose a known risk of contamination, which would indicate that many companies only routinely test for approximately 4 of the primary allergens. All 7 of these surveyed companies only use accredited testing labs, with 4 of them additionally specifying a requirement for ISO 17025 accreditation. In terms of the testing methods employed, all 7 companies predominantly specified the use of ELISA. With respect to defining threshold limits for acceptance, 5 of the companies indicated that they utilise Vital, 1 employed a ppm calculator, and 1 didn't set a threshold when responding to the presence of allergens. The survey also examined the main challenges the respondents had experienced with allergen testing and additional services they felt may be beneficial to them. In terms of concerns, industry-wide guidance on detection thresholds and action limits for each allergen was ranked highly. In addition, labelling specification, what should be tested, how much, and at what point in the supply chain testing should begin were also highlighted. Additional problems identified included detection of cross-reactivity, the effect of processing on test kit performance, difficult matrices to analyse, such as spice mixes and chocolate, as well as the need for more RMs. The requirement for results to be quantified for comparison to vital limits was also highlighted, as results are not always quantifiable, especially those obtained using PCR. Additionally, validation of line cleaning procedures are not always specified, and testing cleaning lines for the presence of allergens may not accurately represent what is in the final product. A final point raised was that numerous companies are looking for more cost-effective, rapid approaches for testing, particularly in terms of point-of-test capability. Allergen testing quality assurance primarily involves the use of certified reference materials, CRMs. The use of CRMs allows for the production of consistent results as well as comparable allergen analysis between laboratories, and thus provides confidence in the results being generated. CRMs are provided with documentation specifying known certainties for composition. However, as kit performances differ depending on sample matrix type and whether a sample has been spiked with a CRM before or after processing, it is important to ensure a range of reference materials, RMs, are available for each allergen and in both raw and incurred matrices. In practical terms, the time and expense required to undertake this course of action makes it unfeasible. Consequentially, quality control materials, QCs, of known allergen content may be used instead. When determining the presence of an allergen following an incident, multiple methods should be used to accommodate the effect that processing may have on the detectability of allergenic proteins. The ideal workflow for the detection of allergenic proteins begins with an ELISA test that can detect allergens. If a negative result is returned, peptide LC-MS should then be used as a confirmatory step. If this is also negative, further ELISA tests targeting different allergenic proteins should be carried out. In most cases, PCR is not the preferred choice, as it does not directly detect the allergenic protein. However, if it is the only option available, then 2 tests should be undertaken with each targeting a different DNA sequence. Testing should be carried out by an ISO 17025 accredited laboratory using validated methods, run in duplicate, and ideally alongside both a CRM and blank. Finally, the data should be reported in milligram allergenic protein per kilogram of sampled food. To summarize, ELISA is the primary approach adopted for the detection of most types of allergen. It is a well-characterized approach that is both efficient and highly sensitive. However, the lack of information provided with many limits their usefulness and makes kit selection difficult. As a general requirement, data on a kit's LOD, LOQ matrices used for validation, known cross-reactives, conversion factors, and target proteins should be provided as standard. In some circumstances, PCR-based approaches can be better for processed foods due to the relative stability of DNA compared to some target proteins, but it is not an appropriate method for all of the primary allergens. Methods based on mass spectrometry are promising development, with demonstrable high levels of specificity possible. However, further research will be required to ensure the methods are both validated and fit for commercial applications. An open-access centralized databases for allergen testing issues, including cross-reactives and matrix suitability, would be beneficial globally. In addition, industry-wide agreement on allergen threshold levels, what should be tested, how much and when, would provide clarity to all parties involved. Finally, the need for more controlled auditing throughout the supply chain and a focus on establishing CRMs and RMs for incurred matrices for all allergens would be beneficial. This e-seminar has been produced as part of the Joint Knowledge Transfer Framework for Food Standards and Food Safety Analysis, and which is funded by the Department for Environment, Food and Rural Affairs, the Government Chemist Programme for 2023 to 2026, the Department for Science, Innovation and Technology, the Food Standards Agency, and Food Standards Scotland.